Welcome to Biotech Genie, where biology isn't boring, it's a story you will never forget. Hey ninth graders, science doesn't have to be hard or boring. At Biotech Genie, we turn your textbook into an adventure. So, whether it's a cell, DNA, or body systems, let's break it down into a story style. Today, we begin with one of the most fascinating journey in biology. We are going to take a peek into microscopic world inside every living being. Yes, even you, even me. This chapter is called Cell, the fundamental unit of life. Now, let me ask you, what do bricks do for a building? They build it up, brick by brick, right? They are the smallest unit of building. Similarly, this cell is the smallest unit that builds life itself. Before we start, let's pause and think for a second. What if this cell had never been discovered? Any guesses on how that would affect us? How would that affect our understanding of our body? Would biology still exist as we know it? Think about it for a moment. Without the discovery of cell, we would be completely in the dark about genetics, diseases, and even how life works. We wouldn't know how we grow, fall sick, or even heal. No vaccines, no COVID tests, and even those fancy toothpaste ads claiming to protect our cells. Gone. But do you think all of this happened overnight? Not at all. It was a long, winding journey, one traveled by many curious minds across centuries. So now, let's do a little time travel to meet the minds behind this discovery. Back in the late 15th, a Dutch boy named Zacharias Janssen was helping his father, a spectacle marker. One day, while playing with lenses, he stacked together and suddenly everything underneath looked much bigger. Without knowing it, he had created the first compound microscope, the tool that would unlock the hidden world of cells. Then, in 1665, a curious scientist named Robert Hooke used a microscope to look at a thin slice of cork. What he saw surprised him: tiny, box like compartments, neatly arranged like a honeycomb. They reminded him of a small room monks lived in, called cellula in Latin. So he named them cells. Fun twist: he was actually looking at a plant tissue, dead, that was actually dead. So all he saw were the empty walls, not all the living parts inside. A few years after Hooke, in 1674, a Dutch tradesman named Anton van Leeuwenhoek, a man with no formal science training, built a powerful microscope out of sheer curiosity. When he peered into a drop of pond water, he saw something no one had ever seen before: tiny moving creatures. He called them animalcules. Today we know them as bacteria and protozoa. He even examined blood, sperm, and plaque from his own teeth, becoming the first human ever to see living cells. After 2000 years, Hooke in 1831, a Scottish botanist named Robert Brown noticed something curious while studying cells of plants like orchids under a microscope. He observed that inside each cell a small, dark, round body right at the center. That structure he named was nucleus. He didn't know everything it did yet, but he had discovered what we now call the control center of the cell. After Brown's discovery, scientists were buzzing with questions. All living things are made up of cells. Are there rules behind how cell works, in late 1830s, two scientists, Skelden and Schwann, a zoologist, teamed up. They observed that all plants and all animals were made up of cells. Together, they proposed the cell theory. Then came the Rudolf Virchow, who added a powerful idea in 1855. All cells come from pre existing cells, and just like that, the foundation of modern biology was laid. Not long after the cell theory was proposed, another scientist joined the story. That was Johnson Perkinje in 1839. While examining the animal cells, he noticed that there weren't just empty spaces within the nucleus floating in the middle, they were filled with a thick jelly like fluid. He gave the internal substance name protoplasm. This helped scientists realize that cells were empty boxes but active living units full of life. Now we will fast forward to 1953. Two scientists, James Watson and Francis Crick, made one of the biggest discoveries in the biology. They revealed the structure of DNA, a twisted ladder called the double helix. With this discovery, they unlocked the blueprint of life, explaining how traits are inherited, how proteins are made, and how cells pass on information. This cell theory, given by Sheldon and Schwann and Rudolf Virchow. First, the all living organisms are made up of cells. What is this means? That means whether it's a microscopic bacterium or a large animal like a whale, every living being is made up of one or more cells. Unicellular organisms like amoeba or bacteria have just one cell that performs all life function. And multicellular organisms like plant, animals and humans are made of many cells that specialize in different tasks. The cell is the basic structural and functional unit of life. Structural unit means that cells from the body structure, like bricks in the building, and functional unit means that all the essential processes of life, like nutrition, respiration, excretion, function, reproduction, happens inside cells. So, cells don't just build the body, they keep it running. And third, all the cells arise from pre existing cells. It means that new cells are not created randomly or from non living matter. Instead, they are formed by the division of cells that already exist. This concept is essential to understanding the growth, healing, reproduction in all the organisms. Now that we have learned about the history of cells, we move on to the cells themselves. Let's begin with the simplest kind of cell, that is the unicellular organisms. Uni means one. These cells are living beings made of just a single cell. That one cell does everything, every function of cells. It eats, breathes, digests, moves, and even reproduces, all on its own. Examples of unicellular organisms is amoeba, paramecium, and even bacteria. Even though they have a simple structure, they can perform all the function needed to stay alive, like tiny self-sufficient machines. Now, let's talk about the more complex forms of life. These are the multicellular organisms, most of the plants and animals. Multi means many cells. They contain many cells that have different functions. So these living beings are made up of millions or trillions of cells. But here's the coolest part, not all the cells are doing the same function. There's division of labor. Each type of cell has a specialized job. So your body isn't one cell doing everything. It's a team of 37 cells, 37 trillion cells working together to keep you alive. So, let's peek inside a typical plant and animal cell and meet all the organisms. Are you ready? So, the nucleus. The nucleus is the brain of the cell. It controls all the activities and stores the genetic material. And the yellow portion, which is present inside the nucleus, that is nucleolus, that is the factory that builds ribosomes. Next comes the mitochondria. We called it as a powerhouse of cell. They generate ATP via respiration and gives you energy. The next is the Golgi apparatus or Golgi body. The Golgi apparatus modifies, packages and ships proteins and lipids. It's like a FedEx for the cell. Next one is the ribosome. Do you know what ribosomes do? Ribosomes are the protein factories. Ribosomes made proteins from the DNA sequence. They build proteins from amino acids. They are usually found on endoplasmic reticulum or in free cytoplasm. The next one is the endoplasmic reticulum. Endoplasmic reticulum is the transport materials that has two types, rough endoplasmic reticulum and smooth endoplasmic reticulum. So rough endoplasmic reticulum has ribosomes on its outer structure and that makes proteins, and smooth endoplasmic reticulum don't have ribosomes and it helps in making the lipids and detoxifies the poisons from the body. The next one is the chloroplast. Chloroplast is found in plant cells and it is the specialized cell which is used for which is the site for photosynthesis that uses chlorophyll. Okay, so plants have an external outer wall, the cell wall. It's an armor made up of cellulose. It gives shape, support, and prevents bursting. Inside is the plasma membrane, the security guard, a flexible and semi-permeable membrane. It's a picky and only it lets in approved substances. That is a semi-permeable membrane. Did you know that plant cells have both the cell wall and plasma membrane to provide structure and protection, while the animal cells have only plasma membrane because they need a flexibility substance for the movement. Now we move on to the simple processes that takes place inside a cell. So first one is the diffusion and second is the osmosis. Both the terms are similar, but there is a minute difference. The first, diffusion, it is the movement of gases or solute from high concentration to low concentration, like perfume spreading in a room. And second, the osmosis, that is the uh, movement of movement from high concentration to low concentration, but that is in the case of water and only through a semi permeable membrane. Examples: when you soak a resin in water, it gets swells, right? That's osmosis. CO2 leaves the cell and O2 gets inside the cell. So here are three terms. What is hypotonic, isotonic and hypertonic solution? Okay. So first we will see isotonic solution. So isotonic solution means water moves and moves in and out equally, as there is equal amount of water inside the cell and outside the cell. The cell stays normal in the shape and everything of the cell stays in normal condition. That is the perfect balance, like your body's comfort zone. Next is the hypotonic solution. There is more water outside the cell, so it enters into the cell and the cell gets swells and burst. Might burst in some cases, like animal cells, like a resin soaking up water and puffing up. And next is the hypertonic solution, where this cell contains more water than the outside. So the water inside the cell, it leaves outside. The cell gets shrinks and undergoes plasmolysis. This is in case of plant cells, like cells drying out. So here the life cycle of cell. You know, cells don't just sit around, they grow and die just like us. Let's take a peek at their life cycle. Okay, so to get a clearer understanding, what you need to know that life cycle of cell cycle has 4 stages: G1 phase, S phase, G2 phase and M phase. G1 phase, that is where the cell grows and performs normal function. S phase, this is the synthetic phase. DNA is copied in preparation for the cell division. G2 phase, where the cell prepares proteins and organelles for mitosis. And M phase, which is the mitotic phase, that where the cell divides. Nucleus divides, and cytokinesis, where the cytoplasm divides. This is how you grow taller, heal wounds and replace old cells. So yes, you are not the same you as last year. Most of your cells have already been replaced. There are 2 main types of cell division and they have very different jobs. Think of mitosis like photocopying a page. One parent cell divides into identical daughter cells, occurs in body somatic cells and is used for growth, repair and replacing the cells. And the meiosis, it has total 8 stages. Okay, so it is like shuffling a toe desk of genes instead of cards and making 4 random hands. Each one is unique. In mitosis, it divides into only 2 daughter cells, and in meiosis the cell gets divided into 4 daughter cells. This stage, prophase 1, metaphase 1, anaphase 1, telophase 1, which is called as meiosis 1, and from prophase 2 to telophase 2, this is called as meiosis 2, where the cell gets divided into 4 haploid daughter cells. Now, isn't that a lot going on inside a teeny tiny cell? Let's wrap it up. All living things are made up of cells. Cells have specialized organelles. They grow, divide and keep life going. So, next time you get a scratch, remember your cells are already hard at work patching you up. So, okay, catch us on Instagram to hear cells and tissues spill their secrets. Join our Telegram for quizzes, MCQ and memes. We have notes dropping soon for every student. Like, share and subscribe to level up your bio game.